I'm Dot.
I'm Tanya.
And to begin, just to describe how transformation basically works, there are three. What am I gonna say? Y'all. There are three main ways bacteria can receive genetic information. These are transduction, conjugation, and transformation. Genetic transformation is the alteration of a bacterium through the incorporation of a plasmid into its DNA. A plasmid is a small molecule of DNA that is cut by restriction enzymes and connected to the bacterium's original DNA. Restriction enzymes cut plasmid sections to form sticky or blunt ends. To connect with the original DNA, a plasmid must form sticky ends and use DNA ligase to fuse the two strands together. The plasmid DNA is then incorporated into the bacterium's genetic information.
So, antibiotic resistance is when bacteria develop the ability to survive the exposure to antibiotics that had killed them or stopped their growth before. Antibiotics work in various ways, including destroying the bacterial cell wall or stopping the bacteria's ability to generate energy from glucose. After bacteria become resistant, these mechanisms no longer work to kill or hinder growth, and the bacteria continue to grow and reproduce in the host.
And this is our pglo satellite colony lab.
We will now talk about the satellite colony lab that we designed. These are pictures of our plates from our original experiment, and the one where we tried to grow satellite colonies was this one. And it's also right here, except much older. The plate that we got our satellite colonies from was this one that had LB, ampicillin, and arabinose with the bacterial colonies that you see here that are very visible. Those are the mutated antibiotic resistant colonies, but then on the outskirts of them they grew satellite colonies that did not glow at all.
In this lab, we were tasked with designing our own experiment, and so for our group, we took satellite colonies from the LB amp ARA plate from the original experiment, and we plated those on an LB amp plate. Our prediction was that those satellite colonies would retain the antibiotic resistance from the original bacteria and would grow in the presence of ampicillin. Our reasoning for our hypothesis was that the satellite colonies would have retained the pglo gene that provided the antibiotic resistance from the original bacterial colonies.
Okay. So there was no growth on the plated satellite colonies on our LB amp plate, and the reason for this is that the ampicillin effectively killed all the cells.
So the explanation for our results of no growth was that the satellite colonies did not inherit or retain the pglo gene from the bacterial colonies that were originally plated.
Okay, so after further research, we learned that the reason that the satellite colonies grew was because the original colonies that did have the pglo gene actually exhibited beta-lactamase, which degraded the ampicillin in the vicinity of the original colony, and this allowed for the satellite colonies to grow without the altered gene.